Hello everyone, welcome to my YouTube channel Biologycolony. In this video, we are going to discuss GATE previous year question of 2023 Excel Zoology, that is Life Sciences Zoology. So let's start with the PYQ discussion. This first question is an MCQ and it carries 1 mark. So which one of the following animals has book lungs as a respiratory organ? Earthworm, scorpion, octopus or starfish? First of all, you need to remember to which group or family or class these organisms belong to. First of all, earthworm, it is an annelid, it belongs to Annelida, and scorpion, it is a arachnida class, it belongs to arachnida class of arthropods. Then we have octopus, it is a mollusk, and starfish, it is a echinoderm. So annelids, they breathe by the help of moist cuticle. So it is cuticle as a respiratory organ. We have octopus, they use gills. Then starfish, they have dermal, branchiae or tube feet. So and scorpions have book lungs. So the answer is scorpions. You can see here, this is the diagram of the book lungs of scorpion. This table is important. You must learn all the animals with their respiratory organs. So the next question is, which one of the following describes the innate behavior of an animal? First of all, innate means from inside. So the behavior which is already present in an organism, it comes from within without even teaching it. It is a innate behavior. So the options are a behavior that is triggered due to the change in environment. No, environment does not affect the innate behavior. A behavior that is trained by the parents, not at all. A behavior that is determined by heredity. Yes, it is a correct answer. Then the next option is a behavior that is learned by hit and trial approach. No. Next question is, which one of the following represents a true ecological population? A pitcher plant and a trapped fly in it? All animals that live near each other in a national park? The leeches and the flatworms that live in a forest? Or all the lions in a reserve forest? So the answer will be all the lions in a reserve forest. Why? Because if we see the definition of population, a population is a subset of individuals of one species that occupies a particular geographic area and an sexually reproducing species interbreeds. So these key points, that is, a population is a subset of individual of one species. So all lions, they belong to one species. Okay, then they must interbreed. So all the lions must interbreed. So the answer is all lions in a forest. Next question. Which of the following animals show bottle cells during the gastrulation stage of development? Snails, amphibians, birds, mammals. So the answer is amphibians. So the bottle cells, these are found in amphibian embryo. This image is given, it represents xenopus embryo. So these bottle cells invaginate to form the slit-like blastopore. These bottle cells change their shape dramatically. They initiate the formation of archenteron. As you can see here, these are the bottle cells and the archenteron is formed by the invagination of these bottle cells. Although experimentally removing the bottle cells does not affect the involution of the deep or superficial marginal zone cells into the embryo, removal of these IMZ cells stop formation of the archenteron. So the next question is, the organism that obtains energy from inorganic compounds are known as autotrophs, organotropes, lithotropes or phototropes. So the answer is lithotrophs. If we see the definition of all the four terms, we get an autotroph is an organism that can produce its own food, like plants. An organotroph is an organism that obtains hydrogen or electrons from organic substrates. Then a lithotroph is a microorganism that use inorganic compounds as electron donors to conserve energy for growth. So they use inorganic compounds as electron donors, while phototroph, they use visible light as a primary energy source for metabolism, or process known as photosynthesis. So in plants. Next question is, which one of the following is or are the causative agents of filariasis? Now here we can see is or are is given. It is a MSQ. It is a multiple select question. So Wuchereria bancrofti, Leishmania donovani, Brugia malai or Trypanosoma gambians. So the correct answer will be Wuchereria bancrofti and Brugia malai. Wuchereria bancrofti and Brugia malai cause filariasis or lymphatic filariasis. Leishmania causes Kala Azar. It is also known as Leishmaniasis. Then Trypanosoma gambians causes African sleeping sickness. Sleeping sickness. The causative agents of lymphatic filariasis include the mosquito-borne filarial nematodes that are Wuchereria bancrofti, Brugia malai and Brugia timori. An estimated 90% of LF cases are caused by Wuchereria bancrofti. It is also known as Bancroftian filariasis. While Leishmania donovani causes a disease called Leishmaniasis. It is a human blood parasite responsible for visceral Leishmaniasis or Kala Azar. Then we have West African sleeping sickness that is caused by the Trypanosoma brucei gambians. It is a long-term infection which can last for years. So this question is numerical answer type question. It carries 1 mark. In a population of 1000 wild dogs in a grassland, 360 and 480 dogs had black body color with genotypes B and B small b respectively. In the same population, remaining dogs were white in color with a genotype of small b. So based on this data, the frequency of allele B in the population is. So how to calculate it? First of all, it is based on the Hardy-Weinberg law. We are given the total number of dogs as 1000, out of which हमारे पास है ब्लैक डॉग्स एंड वाइट डॉग्स. ब्लैक में हमारे पास कैपिटल बी एंड कैपिटल बी स्मॉल बी जीनोटाइप्स हैं, हैविंग 360 प्लस 480. Okay, so the total is 840. Now can we calculate the number of white dogs that is having the small b genotype? Yes, how? 1000 minus 840. तो कितना आ रहा है? We are having 160 white dogs. Now we have to calculate the frequency of allele B, small b. It can be calculated as frequency of small b plus half of the frequency of heterozygote, that is capital B small b, divided by total dogs or total individuals, that is 1000. So 160 plus 480 divided by 2 upon 1000, that is 160 plus 240 divided by 1000. So we are having 400 divided by 1000, that is 0.4. So we are having the frequency of allele B as 0.4. 0.4. Okay. Next question is also NAT and it carries 1 mark. A mature red sperm cell has 2.5 micrograms of genomic DNA, that is equivalent of a haploid genome. Compared to this sperm cell, the amount of genomic DNA in micrograms in a somatic cell which is in the G2 phase of the cell cycle will be. So सबसे पहले हमें पता है कि हैप्लॉइड जीनोम जो है उसका है 2.5 माइक्रोग्राम्स और ये क्या है? स्पर्म सेल, दैट इज इट इज अ गैमीट. देन जब ये इसका हमें जीनोमिक डीएनए निकालना होगा तो 2n कर दिया जाएगा हैप्लॉइड डिप्लॉइड, so it will make 5 micrograms. Now when it goes to G2 phase, अब G2 फेज में जब जाएगा तो वो S फेज से होता हुआ जाएगा, एंड S फेज में क्या होता है, डबल हो जाता है अमाउंट, ठीक है, 2 टाइम्स. सो G2 फेज में जो जीनोमिक DNA का अमाउंट होगा दैट विल बी 10 micrograms. Now 2 marks questions have started. So the first question is, in an experiment, excess amount of bicoid mRNA more than wild type expression level was injected into the posterior pole of a wild type Drosophila embryo at pre-blastodermal stage. Out of the following options, which one represents the best expected phenotype in the resulted developing embryo? Normal embryo with head structure at anterior and tail structure at posterior pole. Head structure only at posterior pole of the body. Tail structure at anterior and head structure at posterior poles of the embryo, while head structure at both anterior and posterior poles of the embryo. So the answer will be head structure at both posterior and anterior poles of the embryo. That is 2 heads formation. Why? As we know, in normal embryonic development, anterior portion contains high concentration of bicoid mRNA, which leads to the formation of head, and at the posterior pole, tail is formed because low concentration of bicoid mRNA is there, or nil concentration is there. Then these are the mutation cases, that is if development of bicoid deficient mutant, अगर bicoid है ही नहीं तो फिर क्या होगा, 2 टेल बनेंगी, क्योंकि एंटीरियर में भी बाइकॉइड नहीं है तो हेड नहीं बनेगा, सिमिलरली पोस्टीरियर तो टेल ही रहेगा. देन अगर बाइकॉइड एमआरएनए को एम्ब्रियो के किसी स्टेज पे किसी पोर्शन में ऐड करते हैं, फॉर एग्जांपल एंटीरियर एंड पे ऐड किया बाइकॉइड म्यूटेंट, मतलब जिसके पास बाइकॉइड एमआरएनए नहीं है, उसमें हमने एंटीरियर पोर्शन पे ऐड किया तो नॉर्मल डेवलपमेंट हुआ, ठीक है. नाउ व्हेन वी एडेड इट इनटू द मिडिल पोर्शन में जैसे ही डाला तो टेल या फिर हेड इन द मिडिल फॉर्मेशन हुआ. देन जैसा कि इस क्वेश्चन में बोला गया है, जब पोस्टीरियर साइड पे डाला गया वाइल्ड टाइप एम्ब्रियो के, मतलब इसमें बाइकॉइड एमआरएनए है पहले से, ये BCD प्लस है, ठीक है, जो कि एंटीरियर पोर्शन पे प्रेजेंट है. सो नॉर्मल हेड फॉर्मेशन यहाँ से हो रहा है. देन यहाँ पर जो पोस्टीरियर एरिया है वहाँ पर भी हेड फॉर्मेशन हुआ, क्योंकि यहाँ पे बाइकॉइड एमआरएनए इंजेक्ट किया गया है. सो टेल फॉर्मेशन कहाँ हो रहा है, एट द मिडिल पोर्शन. सो हेड टेल हेड या फिर 2 heads formation is seen. So the next question is, it is also of 2 marks. Match the hormone/precursor listed in column 1 with their chemical type in column 2 and the tissue of origin listed in column 3. So these are the hormones given in column 1. Glucagon. So glucagon is a peptide hormone and it is secreted by alpha cells of the pancreas. So next is pregnenolone, and it is a steroid hormone and it is secreted by adrenal glands. Then we have FSH, or follicle stimulating hormone. It is a glycoprotein hormone and it is secreted by anterior pituitary. Then we have melatonin. Melatonin is derived from tryptophan and it is secreted by pineal gland present in the brain. So option A is correct. You can see the following explanation. Glucagon is a peptide hormone produced by alpha cells of the pancreas. It raises the concentration of glucose and fatty acids in the bloodstream and is considered to be the main catabolic hormone of the body. Then pregnenolone is a steroid hormone naturally produced in the body by the adrenal glands. Pregnenolone is also made from cholesterol, so it is cholesterol derived and is the starting material or precursor in the production of testosterone, progesterone, cortisol, estrogen and other hormones. These all are steroid hormones. Because the body uses it biochemically as starting material, pregnenolone is sometimes referred to as precursor or prohormone. It displays neuroactive properties, that is, it enhances learning, memory and myelination. Then we have FSH, or follicle stimulating hormone. It is a gonadotropin. Then we have it is a glycoprotein polypeptide hormone. It is synthesized and secreted by gonadotrophic cells of the anterior pituitary gland and regulates the development, growth, and pubertal maturation and reproductive processes of the body. Next is melatonin. Melatonin is a natural hormone that's mainly produced by the pineal gland, which is a tiny gland in brain. It is derived from tryptophan. As you can see in the structure, the chemical structure of both are very similar, and tryptophan Next question is, match the syndromes listed in column 1 with the cause/symptoms listed in column 2. First is Prader-Willi syndrome. It is a genetic disorder usually caused by the deletion of a part of chromosome 15. Then we have Down syndrome. Down syndrome occurs by the third copy of chromosome 21, presence of third copy of chromosome 21 or trisomy of 21. Then we have Cushing syndrome. It is a collection of signs and symptoms due to prolonged exposure to corticosteroids like cortisol. Then we have Turner syndrome. Turner syndrome is a genetic condition in which a female has partially or completely missing X chromosome, that is XO condition. Then we have Fanconi syndrome. Fanconi syndrome is a syndrome of inadequate reabsorption in the proximal renal tubule of the kidney. So option C is correct. Next we have match the immunological statements in column 1 with the appropriate descriptions from column 2. So active acquired immunity. It is a direct contact with pathogens that enter the body. Then we have first line of defense, that is surface barriers like skin. Then passive natural immunity. It is the antibodies passed through the placenta. Then second line of defense is complement proteins and interferons. So option A is correct. Next question is, match the standard/stated cofactors in column 1 with the respective enzymes in column 2. Copper. We have copper, that is the cofactor for enzyme cytochrome oxidase. Then selenium is for glutathione peroxidase. Nickel for urease, and potassium for pyruvate kinase, and molybdenum for dinitrogenase. So option B is correct. You can also refer to this table. These are important for GATE point of view. Here enzymes and cofactors are given, and also enzymes and their coenzymes are given. So you must remember it. Now this is an MSQ. As you can see, the presence of excess glucose has been known to prevent the induction of lac operon as well as other operon controlling enzymes involved in carbohydrate metabolism in E. coli. Which of the following processes defines the phenomenon? First of all, as we know, as glucose level increases, cyclic AMP level decreases. Also, if glucose decreases, cyclic AMP level increases. And if cyclic AMP level increases, what happens? It binds to the CRP, and CRP and CAMP complex activates the it forms the cap complex, which binds to the RNA polymerase. Okay, then this leads to the transcription of beta galactosidase, and thus it activates the lac operon. So when glucose level is high and CAMP level is low, this complex formation will not occur, thus leads to the repression or the blocking of the beta galactosidase synthesis. Thus it is known as catabolite repression. Here glucose is acting as a catabolite, and it also shows the glucose effect. So A and C option both are correct. Now this question is also multiple select question. It is a MSQ. Which of the following techniques are used for determining the three-dimensional structure of proteins? Now, cryo-electron microscopy, yes, it also determines the 3D structure of proteins. Circular dichroism, no. NMR, yes. X-ray diffraction, yes. It is also an MSQ. Among the following statements, which is or are true regarding the replication of DNA? First is replication is bidirectional, yes, and conservative in nature, no. It is semi-conservative in nature. Then we have replication in eukaryotes takes place at multiple ORI sites or ORI sites simultaneously, yes, it is correct. Both the strands replicate in discontinuous manner. Both नहीं करती, एक continuous और एक discontinuous से करती है. So D is correct. A and C are wrong. So B and D. Now the next question is, which of the following statement is or are true for colchicine? It is also a MSQ question, multiple select question. It carries 2 marks. Option A is, it binds to tubulin molecule and disrupts the assembly / polymerization of microtubule. Yes, it is correct. True. It inhibits crossover of chromosomes during meiosis. No, it plays no such role. It inhibits chromosome condensation during prophase. No, but it blocks mitotic cells in metaphase. Yes, it is true. So A and D option are correct. So the next question is from genetics. Wild type Drosophila females having 3 linked genes capital A, capital B, capital C, that is all dominant, were crossed with triple recessive mutant small a, small b, and small c males. The F1 female progenies capital A, capital B, capital C, small c were backcrossed with the triple negative mutant recessive males. The cross resulted in following number of progenies in F2. So these are the progenies given and the total were 1000. The order of the gene as determined from the above data was found to be A, B, C. The recombination map distance in centimorgan between A to C is, we have to calculate the recombination map distance in centimorgan between A to C. So first of all, we have to determine which one are the parental types. So higher the frequency, जिसकी हाई फ्रीक्वेंसी होगी वो पेरेंटल होंगे. तो फिर कौन से पेरेंटल हैं? We have 241 and 252. These two are the parental type. Then next step is to identify the double crossover, that is DCO. So DCOs are the lowest frequency members. These are 14 and 17. Next we have to identify single crossovers. As the order between gene is A, B, C, so first we have to calculate the frequency between A and B, then between B and C, or the map distance between A and B and B and C, then we have to add them to get the distance between A and C. Okay, so the single crossovers between A and B, how can we calculate them? We have to calculate them from the recombinant types. A and B के बीच में आप देख सकते हैं small a और capital B small b है. तो ऐसा ही सेम चीज़ हमें ढूंढनी है बाकी जो सिंगल क्रॉसओवर्स में. कहीं दिख रहा है? यस, इधर है. देन सिमिलरली दूसरे डबल क्रॉसओवर में भी हमें देखना है. तो कैपिटल ए, स्मॉल बी, वी हैव टू फाइंड सेम इन दिस वन. सो these two are the single crossover between A and B. Similarly here, and these are the DCO and these are the parental, and the other one is here. Okay, now the next step is to calculate the single crossover between B and C. We have to identify them with the help of the double crossover. So here we can see capital B, small c. capital B, small c, small c. This is the SCO between B and C. Similarly, SCO between B and C. Okay. Now the formula is to add the number of SCOs and DCOs upon total, and if we have to have them in centimorgan, we have to multiply by 100. So for A and B, we can calculate it as 112 plus 103 plus 14 plus 17 divided by 1000 and multiply by 100. So we have 24.6 centimorgan. If you calculate this, you can get the same. फिर अब B and C के बीच में देखना है तो कौन से है 134 plus 127 plus 14 plus 17 divided by 1000 multiply by 100. ओके, तो ये आएगा 29.2 सेंटीमॉर्गन. अब जब हम इनको ऐड कर देंगे तो कितना आएगा 53.8 सेंटीमॉर्गन. ठीक है, तो ये हमारा आंसर. द रिकॉम्बिनेशन मैप डिस्टेंस बिटवीन A and C is 53.8 centimorgan. Detailed explanation next page पे दे रखी है. You can write it. Okay, so the next question is numerical answer type question. The length of a double helical DNA molecule is 13.6 kilometers. If the DNA double helix weighs 1 into 10 to the power minus 18 grams per 1000 nucleotide pairs and rise per base pair is 3.4 angstrom, then weight of the double helical DNA molecule in nanograms will be. First of all, हमें number of base pairs निकालना पड़ेगा. तो number of base pairs निकालने के लिए हमें लेंथ दे रखी है डीएनए की, दैट इज 13.6 किलोमीटर, और राइज दे रखा है 3.4 एंगस्ट्रॉम. आप देख सकते हैं इनकी यूनिट अलग है, तो हमें एंगस्ट्रॉम को किलोमीटर में करना है. तो कितना हो जाएगा 13.6 किलोमीटर डिवाइडेड बाय 3.4 इंटू 10 की पावर माइनस 13 किलोमीटर्स. ये कैसे किया? हमें पता है 1 एंगस्ट्रॉम इज इक्वल टू 10 टू द पावर माइनस 10 मीटर्स. तो फिर 3.4 angstrom is equal to 3.4 multiplied by 10 to the power minus 10 meters. अब मीटर को किलोमीटर में करने के लिए हमें पता है डिवाइडेड बाय 1000, तो हम इंटू 10 की पावर माइनस 3 और कर सकते हैं. सो 3.4 मल्टीप्लाइड बाय 10 टू द पावर माइनस 13 किलोमीटर्स. तो ये हो गया यूनिट कन्वर्जन का फॉर्मूला. ओके, तो ये हमें कितना दे रहा है 4 मल्टीप्लाइड बाय 10 टू द पावर 13 बेस पेयर्स. ये निकल गया नंबर ऑफ बेस पेयर्स. अब क्वेश्चन में दे रखा है कि जो वेट है इट इज पर 1000 न्यूक्लियोटाइड पेयर्स. तो अब पर 1000 न्यूक्लियोटाइड पेयर्स करने के लिए हमें इसको 1000 से डिवाइड करना पड़ेगा. तो कितना हो जाएगा 4 मल्टीप्लाइड बाय 10 टू द पावर 10 बेस पेयर्स. देन वेट ऑफ द डबल हेलिकल डीएनए मॉलिक्यूल क्या होगा? ठीक है, अब हमें वेट निकालना है. तो वेट ऑफ डीएनए मॉलिक्यूल weight of DNA molecule is equal to 4 multiplied by 10 to the power 10 base pair multiplied by 1 into 10 to the power minus 18 grams. तो ये कितना आ रहा है 4 into 10 to the power minus 8 grams. अब इसमें क्वेश्चन में पूछा गया नैनोग्राम्स में चाहिए इनको, तो हम नैनोग्राम्स में कन्वर्जन कर देते हैं. कितना हो जाएगा 4 multiplied by 10 to the power minus 8 into 10 to the power 9. तो फिर it is 40 nanograms. So the answer is the weight of the double helical DNA molecule is 40 nanograms. अभी नैनोग्राम का कन्वर्जन भी देख लीजिए एक बार. तो 1 नैनोग्राम होता है 10 to the power minus 9 ग्राम. ऐसे ही 1 ग्राम हम लिख सकते हैं 10 to the power 9 nanograms. Okay.